Okay, hey everybody, welcome to the week 2 video for Biochemistry 2. I'm going to be sharing my browser here to show you some of the documents that we worked on this past week. I'll start by clicking over to the periodic table just to remind you that we're really trying to understand the table and to build on our understanding of it to understand larger scale phenomena, materials that are available to us, whether naturally, such as amino acids that our proteins are made of and that all living things make proteins from, or whether it's like building materials we could build a building out of, for example. Um, Reminding you that the left side of the periodic table is called the S block, right? And those elements are elements that have either one or two, depending on the column that they're in, valence electrons. Those are the furthest out electrons. So, for example, sodium, I hope you're getting by now, has the same electron configuration as neon, element number 10, which is a nice stable element. There it is over there. But sodium has one extra electron in a neutral atom. So we would typically draw it like this, basically signifying that it has a stable number plus one extra. And we call that one extra its valence electron, its outermost electron. Just reminding you that valence is the same word root as covalent, right? So covalent just means putting your valence electrons together. Talk more about that in a minute. Now, we did go over in class, like, if we have a really large electronegativity difference, such as between sodium and chlorine, for example, if I were to draw out chlorine's valence electrons, that says Cl, then I would draw 7 of them, because again, it is neon, plus it is 1, 2, 3, 4, 5, 6, 7 spaces further up the table than neon. Not quite all the way to argon, very, very close to argon, just needs one more electron to be like argon. And again, sodium, if it could just get rid of this one electron, would be like neon. Both of those configurations would be stable. So one way to say that is that sodium happily donates its electron to chlorine, which chlorine then becomes chloride. We would write the new situation as Na plus and Cl minus. And if we want, we can show Cl minus as 8 valence electrons. And sodium has dropped its one. So now both elements are stable in terms of electrons. Another way we could frame this interaction is that sodium and chlorine have an electronegativity difference of 3.0 minus 0.9 or 2.1. Chlorine very much wants to pick up an electron. It has a very high electronegativity. That's a way of signifying that. Sodium doesn't really want to hold on to its electron. It has a very low electronegativity. That's a way of signifying that. Remember that if we are greater than 2.0 in terms of electronegativity difference, that's going to form ions. So we call this process of electron transfer ionization, and we call the results of it ions. If I were to be more specific, I would point out that sodium, being the positive ion because it has lost an electron, electrons are negative, so subtract a negative, it becomes positive. Any positive ion we call the cation, and any negative ion we call the anion. So chloride here is the anion. Now, once ionization has taken place, it's a relatively permanent situation. Sodium and chlorine are much happier this way. They do not want to, go back to the starting point of sodium having one too many and chlorine having one too few. That'd be a lot less stable. So they basically stay like this. But now that they are like this, we noted that because they have opposite charges, they'll be attracted to each other. So that is referred to as an ionic bond. Cool. We practiced picking elements that would make ionic bonds with each other from the table. And one thing that we arrived at is that it's best to pick something from the metal part of the table, either one of the first two columns, which are the um, alkaline metals. We're going to cut off hydrogen here, actually, because hydrogen is not a metal. So we will do this. That's more accurate. So lithium down to francium are the alkaline metal or alkali metals, and beryllium down through radium are the alkaline earth metals. You could pick from there, or you could pick from the middle, which are the transition metals. And if you're going to pair those up, you'd want to pair them up with non-metals. So those would basically be these. So pairing up a metal with a non-metal will give you usually ionic bonding. Not always, you can find exceptions, but often that's the way to get ionic bonds. Okay, you'll get ionization happening, and then you'll have an attraction between the positive and the negative. So we practiced that a bit. We saw some easier examples like sodium chloride. We also saw some more complicated examples like calcium chloride, for example, where calcium is in the second column. It has 2 electrons it would like to get rid of. And so it transfers both of those to chlorines. It needs 2 chlorines to accept those 2 electrons, so it forms a compound called calcium chloride, but that is CaCl2. The hardest version of this that we did involved barium from the same column as calcium. It has 2 valence electrons, and nitrogen. Nitrogen is in the fifth column out of 8. If you're going across the first and second, or sorry, the second and third rows. And so it needs to pick up 3 electrons in order to gain stability. Well, barium has 2, so not a great situation. So what ends up happening is that 3 bariums and 2 nitrogens will interact. The 3 bariums will transfer 2 electrons each. The 2 nitrogens will accept 3 electrons each, and that means 6 electrons are transferred overall. The ions that are made are Ba2 plus and N3 minus. We would call that barium nitride. And yes, indeed, they will also attract each other because they are opposite charges. Again, to practice some terminology, calcium and barium are cations here. Chloride and nitride are anions. You can tell the naming convention here is to keep the cation's name as the same as the element. So barium stays barium, calcium stays calcium, and so forth. And to make the anion into an ide ending instead of chlorine, chloride, instead of nitrogen, nitride. So that's the naming convention. Now, the other type of bond that we have been studying is a covalent bond. And I pointed out to you all that covalent bonds basically come in 2 flavors. One is the uneven sharing of a polar covalent bond. The other is the even sharing of a nonpolar covalent bond. To figure out which one you've got, it's all about the electronegativity difference. Typically, with covalent bonds, you'll want to bond non-metals with non-metals. Many of you initially picked metals with metals to make these bonds. Metals with metals tend to make different types of bonds called metallic bonds, and because they're more complex, we're not going to study those right now. If you want to look them up, you can look them up, look up metallic bonds, and learn about them. They're sort of strange and sort of awesome, but they are definitely different from the covalent bonds that you find when you've got non-metals binding with non-metals. So again, to focus us on the right part of the table, we're basically talking about this part here and also including hydrogen. I also hearkened back to last year. Fonk are the 5 most common elements in living systems. And you'll note they make covalent bonds with each other, basically. So that's a good place to start. The most significant polar covalent bonds, as you've probably realized by now, are between H and O, and then also between H and N. Those are really common polar bonds. You're now beginning to put together why, which is that there are these functional groups that are located in biological molecules, such as the first one I drew, which is the hydroxyl, OH. The second one that I drew, which is the amine group. Or even this third one, which is the carboxyl. So these types of bonds are common in living systems. And again, I'll just type out the names so that you can see them here for your records. Hydroxyl, um, amine, and carboxyl, or you can call it carboxylic acid. Those are the names of those particular groups. Cool. So those are great bond types to focus on, H with O or H with N, where the O or the N will become the partial negative and the H will become the partial positive in both of those situations. And again, all you have to do is look up at the table. H has a lower electronegativity than N by a large enough difference, zero point 0.9, that the bond is uneven or polar. And with O, even more classically polar, 1.4 difference, which is why water is such a polar molecule, right? Because it's got a really solid difference of electronegativity between the O and H in each of its bonds. Okay, so those are some good examples of polar covalent bonds. Finally, the real classic example, and again, you've now seen this over and over, so I'm hoping it's beginning to make these feel recognizable and a little bit more friendly and less confusing. Nonpolar covalent bonds classically happen between carbon and hydrogen. And if you look back up on the electronegativity table, you find that carbon is sitting at 2.5, hydrogen at 2.1. That difference is 0.4, so solidly in the nonpolar covalent section of the differential. It also kind of goes without saying, but I'll say it, anything bonding with itself is clearly going to be nonpolar. Carbon with carbon, 2.5 with 2.5, that's a zero difference. So that's as nonpolar as it gets. That's as even a sharing as you can achieve. So we practiced this week the application of those covalent bonding principles to these three different amino acids and then to the full chart of 20, which we'll take a look at in a minute. The three samples that I gave you, which I thought were three of the easier ones to understand, not all of them are equivalently easy, and I'll say more on that in a minute. But if you take a look at these amino acid structures, I gave you phenylalanine, asparagine, and glutamate. And I told you the backbone part of each of them is here. We're going to learn more about that backbone in the coming week 3 lessons. It's a really interesting thing that backbone gets involved in doing when you make a protein. And you'll see why the side chains, which are the other parts that I haven't circled in red, why they are so significant and why we focus on whether they are nonpolar, polar, or positively or negatively charged, for example. Okay, so these three examples of variable side chains, the side chain of phenylalanine, asparagine, and glutamate, are kind of case studies in each of the possibilities. Phenylalanine has only carbon-carbon bonds and carbon-hydrogen bonds. Remember that this CH2 thing up at the top here, I did draw this out. That's just a shorthand way of writing this without having to like draw everything out and show all the bonds. So those are also just CH bonds and CC bonds. So every bond here is either CC, which is a difference of 0, nonpolar bond, or CH, there's that classic difference of 0.4, nonpolar bond. So the whole thing is considered just nonpolar. It's not water soluble, it's fat soluble because things that are water soluble are polar, just like water, and things that are fat soluble are nonpolar, just like fats and oils are. The one in the middle contains 3 bonds that have polarity. And again, I'll X out this H here and write out the full amine group. NH2 is an amine group. So there it is, N with H, N with H. So this is a polar bond. This is a polar bond. Those both have an electronegativity difference of 0.9. And then C with O, I think it's around 1. Is that right? Yeah, 2.5 to 3.5. Let's see if I can scroll back to the right spot here. So that would be a difference of 1.0 in this bond. Those are all solidly in the polar electronegativity difference. Now, you could point out that C to N is 0.5 and C to C is 0, and the CH2 thing, again, is like, those are CH bonds, those are 0.4. So there are a handful of nonpolar bonds here too, but 3 polar bonds that kind of dominate the bulk of the side chain. If you look at the amount that I've circled in green here, there's a lot of polar stuff going on. And so overall, we class this as a polar variable side chain. I'm not going to ask you to make difficult judgment calls between polar and non-polar side chains. If I give you one, it'll be either pretty clear that it's got a lot of polar stuff going on, or pretty clear that it has no polar stuff going on. So I won't make you make a judgment call on one of the harder ones if I were to ask you this in any kind of a graded situation. Um, the final one on the right here is glutamate, and that one I'll point out to you is charged because of this carboxyl or carboxylic acid group. One thing that a carboxyl can do, this is why it's called an acid. Are things that can add H pluses to a solution of water. And this carboxylic acid, having added its H plus, that leaves behind an O that is now lonely without its hydrogen. Remember that before it would have looked like this. So it now has no hydrogen with it. And since hydrogen left without its electrons and just left as an H plus, the electrons from the bond get left behind and the O becomes O minus. Okay, so that's why this is written with a minus charge. That is therefore a negatively charged variable side chain, and that makes it an ion. Anything with a full negative or full positive is ionic in character. So you'll find there are two negatively charged side chains, glutamate or glutamine. And if I click over to the full thing here, I guess I might as well show them glutamate and aspartate. And both of them have carboxyl groups, carboxylic acid groups, that when their hydrogen leaves, they're left just C double bond O, single bond O minus. The positively charged R groups all have nitrogens that can pick up an extra H plus and become positive. Okay. And then you see all the others. Most of the nonpolar ones are very straightforward. They're nothing but C's and H's. This one happens to have a sulfur in it. No big deal. It has basically the same electronegativity as carbon. The ones that are polar tend to have hydroxyls on them or amines, right? These H2Ns. There's asparagine that we looked at in the previous slide. There are also some weird ones, like proline is very weird. The side chain actually like loops back and sticks to the backbone, and that makes it behave in like a polar way. That's super weird. We're not going to worry about that. Cysteine, even though sulfur is basically like carbon, there's really no polar bonds in cysteine, but it still can behave in a polar way. Shout out to some folks in the A period class who were asking really great questions about like, well, what the heck is going on with cysteine? And I was actually like, I don't actually totally know. I've seen it listed as nonpolar before. What's going on? So I did a little bit of reading. You're welcome to follow up on this on your own to read this and see that I'm telling you the truth. But if you look up cysteine's polarity or cysteine polar versus nonpolar, it kind of has both characters. It's capable of being classed as both polar and nonpolar. And that's kind of because like this sulfur has some electron density on it that are just like normal valence electrons hanging out. And so it kind of can be induced to have a little bit of a negative character to it just because it has these electrons. It can also not do that. So it's very context dependent. And if you're like, but that's so frustrating, right? So ignore these two in terms of like classic examples. They're wild and weird, and that sometimes is what's going on. But the other 18 are pretty dang straightforward. You've either got totally hydrocarbons, like all of the ones in the top left here. You've got ones with hydroxyls or amines, C double bond O's on them, like the other four in the polar box here. You've got ones that have got a plus charge or a minus charge, or then you've got these like ring structures. And even tyrosine and tryptophan, which do have one polar bond each, it's not quite enough to offset how many carbons. Remember that there's a carbon at each of the points on these rings. How many carbons and how many hydrogens are all over these? Each carbon would have like extra hydrogens just to make it have 4 bonds here. So this is so much hydrocarbon that the one little polar bond at the end of it can't quite do it. Does that still like does that OH, that hydroxyl still have implications for tyrosine and the way that it works? Oh, heck yeah, definitely. Tyrosine's really important in proteins. It's really important that it has that hydroxyl, but it doesn't give it an overall polar character. The more that you learn about these, the more fun that they are. They're really interesting and they do all kinds of interesting things. The next thing that we're going to do now that we've got a basic sense of how to tell nonpolar from polar, and in most cases we're pretty comfortable. I'd say for 18 out of the 20 of these, it's quite sensible and not too hard to look and figure out what's what. Now that we've done that, we're going to have the opportunity to actually model some real proteins and to figure out like why do they take on the very complicated shapes that they take on. The awesome part about this is that you'll realize it's really all just coming down to the polar and nonpolar and positive and negative side chains that they have stuck to them and the order that they are linked together in. We're going to tie this all the way back to last year's material on genetics, and you should be able to really begin to understand why living organisms have the particular traits that they do from a molecular level all the way up through the ecosystems that they end up generating through the interaction of their traits. It's really kind of, I have like goosebumps, chicken skin, whatever you prefer, thinking about that whenever I think about it, because it really gives you a window into the biochemical functioning of the Earth system that we live in. And I think that's really, really, really cool and just an epic thing to get to understand a little bit about. Anyway, that's the lesson video. I hope that I kept this at a reasonable length, and I hope it's useful to you. Looking forward to seeing you in class.